Okay, good morning, all of you. Today we shall continue our topic from yesterday about active transport. Now, basically, in today's class, we will talk very briefly about what is active transport, what are some common definitions like permeability and permeation and being permeable. We will see some examples of the active transport and where are these active transport mechanisms happening in the body. Briefly also we will see about the mechanism how this active transport occurs and what are the various metabolic changes that happens and how it is regulated. Now, transport mechanism is very essential in the body because with this transport systems, the cellular functions are made to happen at an adequate rate. Substances which are needed are provided to the cells inside. Substances which are not needed are sent out from the cell into the circulatory system. Therefore, the cell transport becomes an important part in the normal functioning of the cell and as a body as a whole. Now, when we see the structure of the cell, you will find that the cell membrane, the cell is limited in its activity or its environment by a membrane which is called as cell membrane. There are substances which can pass through the cell membrane very easily. Then there are substances which requires little assistance to enter into the cell. But the membrane which is present across the cell is continuity, and this continuity is the integral part of the function of the cell membrane. Any disruption in the continuity of this cell membrane will allow unwanted substances or toxic substances to enter inside the cell, and important substances can diffuse out from the cell from inside to outside if the integrity of the cell membrane is disturbed. In addition to this, apart from being a continuous structure or a continuous membrane, the cell membrane has got certain channels or in the form of proteins, and these proteins provide certain ways or pathways for communication between the inside of the cell and the outside of the cell. Therefore, one of the important function of the cell membrane is permeability. Now, what do you mean by a cell membrane being permeable? The membrane as such, which is covering the cell, does not allow all the substances to move freely across it. It will allow some substances to move across it in either direction, either from outside to inside or from inside to outside. And this ability of the cell membrane to allow the substances to pass across it is called as permeability. Now, once we know what it means by permeability, let us see some examples. For example, the cell membrane may allow certain substances to move across it like sodium ions, potassium ions, calcium ions, either from outside to inside or from inside to outside. It may also allow certain hormones to move from outside to inside. Say for example, the thyroxine hormone. There is one hormone, thyroid gland. The thyroid gland secretes the thyroxine hormone. The thyroxine hormone, once it enters, is released from the thyroid gland and enters into the circulation. it reaches all the cells, and from the circulation, it passes across the cell membrane and reaches inside the cell where it shows its action. Similarly, you have got another hormone, insulin, secreted by the pancreas, glucagon secreted by the liver. Now, all these hormones, when they are released by the respective endocrine gland, enter into the circulation, and ultimately for them to act, they have to reach that particular target cell, and to reach that particular target cell, they have to pass through the cell membrane or enter into the cell membrane. Similarly, water is another part of important substance which is most commonly transported across the cell. Then we have got certain drugs, medications. Suppose you have got certain pain, you have got injured your hand, or there is some sports injury to the muscle, you develop pain in that particular region. Suppose you have had an injury in the thigh or a muscle pull in the thigh, you have pain in the thighs or in the legs. Now, to reduce that pain, you take some tablet or a painkiller. This painkiller is swallowed or an injection is given. It enters into the circulation. From passing through the circulation, it has to reach the particular target organ wherever the pain is. So, for this drug to act and reduce the pain, it has to enter into the muscle cell. To enter into the muscle cell, it becomes important that it transfers or it enters from the outside of the cell to the inside of the cell, that is outside the skeletal muscle to the inside of the skeletal muscle, so that it can act there and it can help in reduction of the pain. So also when you feel a little dehydrated or when you are thirsty, it means that there is less of water inside the cells, or the extracellular fluid contains of less water. So you drink water. When you drink water, the water is absorbed from the GI tract. It enters into the circulation, and from there it is distributed to different parts of the body, and through transport across the cell membrane, this water enters into the cell, and the water levels or the ICF water level is getting corrected. So therefore it becomes important that the cell membrane allows substances to move across it in by either direction, when it is needed from outside to inside or from inside to outside. So the cell membrane is a bilipid layer, and it has got two layers of lipid. The characteristic feature of the cell membrane is that it can allow lipid soluble substances to transfer, to move across from the ICF into the ECF, or from the ECF into the ICF freely, because these substances are lipid soluble. So all those molecules which are lipid soluble substances can easily pass across the membrane. For example, your thyroid hormone. It's a lipid soluble substance made up of lipid, comprises of fatty acids. Since the membrane can easily allow the transport of fatty acid substances across it, so the thyroid hormone can easily move from outside to inside. Once it is inside the cell, it will show its effects. But that is not all for all the substances. For certain substances, this cell membrane acts as a barrier and does not allow substances to move. So, in between this cell membrane, continuity of the cell membrane is interrupted by the presence of certain proteins. Now, these proteins act as structural proteins and act as gateways for substances to move from one side of the membrane into the other side of the membrane. So, these protein substances are nothing but they are called as channels. So, they form a channel on the membrane, where through these channel it will selectively allow the substances to move from one direction into the other direction. So apart from protein channels, there are certain other channels which are also known as water channels, and these water channels will allow the substances to allow water to move freely across the membrane. Therefore, the cell membrane is impermeable to most of the substances, but it is selective permeable. So permeability means allowing certain substances to move across the membrane. Selective permeability means allowing only some selected substances to move across, and these selective substances may be water soluble or they may be lipid soluble substances. Lipid soluble substances can move across freely, whereas other substances will need the help of these protein channels to move across, and water can move through the water channels. Now with this in background, let us go into detail about our active transport. Now what does active transport means? So now this transport of the substances is basically divided into two: passive transport and active transport. Passive transport is a transport where the substances can move across the cell membrane without using energy, active energy, just because there is a concentration gradient existing from one side of the membrane to the other side of the membrane. Now today let us see about active transport. Now active transport is a transport. If you break down the definition, transport means movement of substances across the membrane. Active transport means such transport or such movement of molecules which is occurring against the concentration gradient is known as active transport. So basically the definition of active transport is movement of substances against a concentration gradient which requires energy is called as active transport. The only difference between facilitated diffusion and active transport is that active transport requires the metabolic energy when exactly the transport is moving is happening. So active transport is always against the concentration gradient. Now these are the two models which are depicted here for the transport across the membrane. Now this is a membrane. If you see the frame of the door is the cell membrane. There is a continuity. This is a protein channel. Now this protein channel acts, this gate or the door acts as a channel where whenever there is an opening up of the gate, it will allow certain substances to move from one side, that is from inside to outside. So this is a channel where this channel is allowing certain substances to move from one side to the other side whenever there is an opening up of the gate. This is one model for transport across the membrane. The other is a carrier. Now what do you mean by carrier? Imagine the structure, the carrier, the diagram which you are seeing is exactly in the cell membrane, in between the cell membrane. Now here this is rotating round and round, where you need to understand that once a molecule comes and attaches to one of the frames of that particular carrier, it activates the breakdown of the ATP. The ATP breaks down into ADP and high energy phosphate bonds are released. Breakdown of this ATP releases some energy. Once that energy is released, it causes the carrier to move or rotate. Once it rotates, the substance is transported from one side of the membrane to the other side of the membrane. Normally, this carrier is stagnant. Say for example, on one end the molecule or a substance comes and binds. Once the binding of a substance takes place on one of the frames of that particular carrier protein, it immediately activates the ATP. The ATP gets activated and gets broken down. Energy is released. That energy is used to rotate or cause a transformational change in the structure. So this transformational change, because of the breakdown of the energy, high phosphate bonds, causes it to rotate. When it rotates, the substance which is present either on the inside is now delivered to the outside. So this is another example where in the cell membrane, which is impermeable to certain substances, a carrier protein helps in transport of the substance from one side of the membrane to the other side of the membrane. So basically these are the carrier proteins. A cell membrane may have only channels on its membrane, or may have only carriers, or may have both channels and carriers, and whenever whatever molecule activates it, when it binds onto that, it stimulates the breakdown of energy and a structural or a conformational change takes place, causing the movement of substance to the other side. Now let us see this active transport. The same thing I have depicted again for a better understanding. There you will find that there is a bilipid layer. The bilipid layer is nothing but the cell membrane. The phospholipid molecules are present. The tail ends are facing towards each other. The head ends are towards the other extreme ends. One head end of the bilipid layer is towards the outside, that is towards the blue part, and the other head, that is the phospholipid layer, is facing towards the inside the cell membrane. In between, you have got a carrier protein which is present in between the cell membrane. Now, this carrier protein, if you see, is a gated carrier protein. It is called as a gate because it's an inverted V shape. The above two ends of the V-shaped gate are touching each other, thus preventing any movement of substances from outside to inside or from inside to outside. Now, if you carefully see that carrier protein, you will find that there is a binding site present on the inside of the membrane. Here you will find that there is a binding site. This is the V-shaped protein. This V-shaped protein has got a gate. This gate is particularly closed. But on the inside here, you will find that there is a binding site. Now this end here is the region of the lower concentration and this end is the higher concentration. Now whenever a substance has to be moved from inside to the outside, let's say for example, the sodium ions which are on the outside more and less on inside, if it has to be transported, so sodium has to be transported from the lower region of lower concentration to a region of higher concentration. So it has to move against the concentration gradient. So because it has to move against the concentration gradient, it becomes an active transport. And it is an active transport because here this movement is requiring energy. So how does this transport takes place? It takes place by the presence of these carrier proteins. This carrier protein is a V-shaped protein. Here you will see that there is a binding site present here. Now, whenever the transported particle from the ICF, this is the inside of the cell, comes and binds on to the binding site of this carrier protein, the binding of this transport particle to the binding site will stimulate the ATP here, this high energy bond which is present. It will result in the breakdown of the phosphate ATP. Energy will be released. This release of energy will cause two things. One, it will suddenly cause the opening up of the gate which is on the outside of the cell membrane, and this energy will see to it that this molecule is pushed from inside to outside. That is, it is pushed from a lower energy area to a higher energy area. That is, from a low concentration area to a high concentration area. Thus, it is an active process. So, what do we understand here? Normally, active transport is by means of carrier protein. It occurs against a concentration gradient. That is, the transported particle moves from a region of lower concentration to a region of higher concentration. And how does this occurs? This occurs by the activation of the active carrier protein. And how does this activation takes place? This activation takes place because once the transported substance comes and binds to the binding site, it causes a breakdown of the phosphate molecules which provides energy. Simultaneously, there is a conformational change or a structural change happening in the carrier protein whereby the inside of the gates close and the outside of the channel carrier protein gets opened, thus pushing the substance from inside to outside. Think I have explained this process very clearly. So this is one example of an active transport. Now let us see how many types of active transports are there. There are basically two types of active transport. One is a primary active transport and the second one is the secondary active transport. Primary active transport is a process that directly uses the energy from the hydrolysis of ATP, and this energy is used to transport substances against the concentration gradient. So basically, the word says active transport. Transport is means across the membranes. Active is utilizing energy. Primary means the breakdown of the energy bond, that is the breakdown of ATP, is utilized to cause a structural change in the carrier protein and push that molecule against the concentration gradient. And what is a transporter here? The transporter is nothing but an enzyme which is called as an Atpase enzyme. This Atpase catalyzes the breakdown of ATP and catalyzes the breakdown of phosphorylates itself, releasing high energy bonds. Now, what are the examples of these primary active transport? Now basically the whole of the active transport is of three types. You will find that one is the P type of transport, which is known as phosphorylation type. Then there is a V type of transport, which is known as a vacuolar type. There is a F type of transport where it is known as there is a gating of gate energy coupling factor is there, and then you have got ABC transporters. So basically the transporters are of four types. We have got the P type of ATPases, we have got the V type, we have got the F type, and the other is the ABC types. Now P type, what does this mean? P type of transporters means they cause phosphorylation of the energy bonds to facilitate or to cause the active transport. So that is a P type of transport. V type of transport is nothing but a vacuolar type of transport where the vacuolar energy is used in the transport of substances, and that comes under the example of calcium ATPase energy. Then we have got the F type of energy bonds, F type of transporters, where the F type of transporters are nothing but a energy coupling factor. So that F factor is an energy coupling factor, and that type of transportation, active transport, is basically taking place in the endoplasmic reticulum, in the plasma membrane, and in the sarcoplasmic reticulum. Then we have got the ATPase transporters. So basically you understand that the transporters are of four types: P type, the V type, the F type, and the ABC. Now let us see what are the examples of active transport which are happening in the body. One example of the active transport is the sodium potassium ATPase. This sodium potassium ATPase is an example of an active transport, and it is a P type of an active transport where phosphorylation of energy bonds is taking place and facilitating the movement of sodium ions. Then we have got calcium ATPase mechanism. This calcium ATPase mechanism is nothing but which is a vacuolar type which is present within the cell membrane, and it facilitates the movement of calcium ions across the gradient, across the membrane. Then we have got the another example of hydrogen potassium ATPase. The hydrogen potassium ATPase is again example where there is a transport of hydrogen ions and potassium ions across the cell membrane, and this is against the concentration gradient. Then we have got transportation of only hydrogen ions or protons, which you will see it in the endoplasmic reticulum and in the Golgi apparatus and in the organelle structural organelles. Now to explain to you these examples briefly, the sodium potassium ATPase is an example of active transport. What does this sodium potassium ATPase do? This sodium potassium ATPase transfers sodium and potassium ions across the cell membrane. Now here it is a little different. This will transfer sodium ions from inside to outside, and simultaneously it will transfer potassium ions from outside to inside. Then the calcium ions. What does the calcium do? Where does it does the transport, active transport? It is basically in the cell. We know that the calcium concentration is inside, outside the cell membrane is high. So calcium is transported across the gradient from inside of the organelles to the outside, so that a concentration of calcium ions is maintained constant. Then another example we have is hydrogen potassium ATPase. Where is this hydrogen potassium ATPase seen? The hydrogen potassium ATPase is basically an example of active transport seen most commonly in two areas of the body. One in the GI tract and other is the nephrons. So what does it do in the stomach? You know that the constant, whenever we take food, for digestive system to be most active in the GI tract, the high concentration of potassium is needed so that the pH is maintained around 2. When you are not eating food or in between the meals, after between, let's say, your breakfast and lunch or lunch and dinner, the pH comes back to 7. But when you have taken a meal or when you have eaten some food, for the food substances to get digested and the enzymes to show the maximal activity, the pH of the stomach has to be around 2. So that means if the pH has to be 2, there has to be a high concentration of hydrogen ions in the stomach. Now this high concentration of hydrogen ions means that the cells of the stomach or the epithelium of the stomach has to secrete more amount of hydrogen ions. Simultaneously, it has to transport potassium ions across the gradient. So this high concentration of hydrogen ions in the stomach is maintained by the hydrogen potassium ATPase, where whenever that enzyme gets stimulated, the hydrogen potassium ATPase breaks down. Breaking down of this will release phosphate ions. The phosphate ions will activate the channel proteins, the carrier proteins, and this will transport the hydrogen ions in one direction against the concentration gradient and potassium ions in the opposite direction against the concentration gradient. The other example of the hydrogen potassium ATPase being active is in the kidneys. We know that in the kidneys there is a tubular fluid. As urine is being formed, it passes through the different areas of the nephron, and in the collecting ducts there is a requirement of a high concentration of hydrogen ions to be maintained for the acidic pH of the urine. So to maintain this acidic pH of the urine, there has to be a more concentration of hydrogen ions. That means there has to be an acidification of the urine. So that urine requires high concentration of H plus ions, and this high concentration of H plus ions and potassium ions is maintained by the hydrogen potassium ATPase. So there is an active transport taking place where the potassium is secreted outside and the hydrogen is reabsorbed from the cellular end into the circulation. So two examples of hydrogen potassium ATPases, one in the GI tract and the other in the nephrons. The sodium potassium ATPase is also a similar example where sodium is transported from inside to outside, potassium is transported from outside to inside, and this is happening at every cell in each cell after a functional aspect is over. Then calcium ions, the calcium ions are also probably most required in the muscle during muscle contraction. So at that time the calcium ATPase system gets activated. Then the other is the proton pump proton H plus ATPase system, where these proton ions are exclusively secreted in high concentration, and this is the place this happens in mostly the organelles of the cell. So either it may happen in the mitochondria or it may happen in the Golgi apparatus or in the lysosomes. So these are the four examples of active transport. Now we will basically read across the sodium potassium pump. Now before we go on to the sodium potassium pump, let me explain to you three different definitions. One is uniport. You have to remember uniport, symport and antiport. What does these three means? Uniport means, as the word suggests, movement of ions in one direction is called as uniport. Symport means movement of two ions is called as symport. Antiport is movement of ions in opposite direction is called as antiport. So uniport, symport, antiport. Uniport means unidirectional. Symport means two substances moving simultaneously in one direction. Antiport means two substances moving in opposite directions. Now let us briefly discuss about the sodium potassium pump. Now to understand this, we have to go back into the concentration of the electrolytes in the ECF and the ICF. If you see the slide there, there is one square box which is nothing but the cell. This cell is nothing but the lined by a cell membrane. There is the intracellular fluid, the outside in the blue is the extracellular fluid. The concentration of the sodium in the inside is around 14 milliequivalents and outside it is around 150 milliequivalents. Simultaneously, if you see the concentration of potassium ions, the concentration of potassium ions in the ECF or in the outside is 5 milliequivalents, whereas inside it is 150 milliequivalents. Now, if you see this, the concentration gradient of sodium is more on the outside than on the inside. Whereas if you see the concentration of potassium ions, the concentration of potassium ions is more in the inside than in the outside. But at some point of time, there is a possibility that the concentration of sodium ions inside may increase and the concentration of potassium ions outside may increase, disturbing this equilibrium, disturbing the internal environment, disturbing the homeostasis. So now to bring back that equilibrium, to bring back that internal environment back to normal, these excess sodium ions which have moved inside has to go out, and those excess potassium ions which have accumulated outside may have to go in. That means there has to be a movement of sodium ions against the concentration gradient, that means from inside to outside, and there has to be a movement of potassium ions against the concentration gradient, that means from ECF to ICF. To facilitate that movement, there is a structure or there is a transporter present which is called as the sodium potassium pump. Now coming on to the sodium potassium pump, the sodium potassium pump is again a carrier protein. It is present in between the cell membrane, and whenever it is activated, it will transport sodium ions, 3 sodium ions from inside to outside and 2 potassium ions from outside to inside. That means it is transporting the sodium ions against the concentration gradient. Simultaneously, it is also transporting potassium ions from outside to inside against the concentration gradient. When this is against the concentration gradient, the energy to be provided by this is by the breakdown of ATP into ADP. That energy is used to push or to move the sodium from one direction and potassium from the outside to the inside. So this is an example of the primary active transport of sodium potassium pump. Now with this energetics, with this basics, we will just go on how does this happen. Now if you see this diagram here, you will find that this is the sodium potassium pump. Here this is having two units, alpha unit and a beta subunit. This is the whole of the cell membrane. In between, this protein, structural protein is present, or a carrier protein is present. This carrier protein is exclusively meant to transfer sodium potassium, and it is known as a sodium potassium Atpase pump. Now it has got two units, alpha unit and a beta subunit. Now if you see this sodium potassium pump will move 3 sodiums from inside to outside and 2 potassium ions from outside to inside. By this time, you would have understood that the concentration of sodium outside is more and sodium inside is less. The concentration of potassium inside is more and the concentration of potassium outside is less. So, both these ions, sodium ions and potassium ions, are moving against the concentration gradient, but they are moving in opposite to each other. Sodium moves from inside to outside, potassium moves from outside to inside. Now let us see the next slide here. Here it is exactly showing how exactly the mechanics is happening. Now here if you see this diagram here, there is the active transport. If you imagine this to be the carrier protein or the sodium potassium Atpase pump, this is the inside, this is the outside, this is the cell membrane, the continuity in between the cell membrane, you have got this sodium potassium pump. Now here in the first step, what is happening is there is a binding site. The sodium ions are coming and binding on the inside of this pump, on the binding site of this pump. As soon as this binding of the sodium ions is taking place inside, the ATPase enzyme is activated here. If you see in this blue lines, once this ATPase enzyme gets activated, it breaks down the ATP and gets phosphorylated and releases high energy bond. Once that high energy bond is released, that is phosphorylation has taken place, the whole of the energy is used to twist this carrier protein. So there is a conformational twist or conformational change and rotation of this carrier protein taking place. Once this channel rotates, it causes the movement of the sodium ions from inside to the outside cell membrane. As soon as the sodium ions are on the outside of the cell membrane, the active sites for potassium gets activated. Once the sites or the binding sites of the potassium becomes activated, they here then now have high affinity of potassium. So potassium will come to bind to this. So as soon as the sodium is getting discharged from this binding sites, the potassium is coming and binding onto its own active site. Once the potassium binds here, there is again a dephosphorylation, that is the phosphate bond gets detached and a sudden snapping or a dephosphorylation causing a conformational change. This conformational change causes it to rotate against completely. So what had turned 180 degrees now gets rotated by another 180 degrees. So a 360 degrees rotation takes place. Once this 360 degrees rotation takes place, the potassium has now come into the inside and it gets liberated or it gets freed from the binding site and into the ECF. Thus you will see here that two things are happening: sodium ions binding, dephosphorylation taking place, a conformational change taking place, 180 degrees rotation of the channel protein taking place. So the sodium which was inside now moves to the outside. As soon as the sodium is moving outside, the affinity of the potassium binding sites increases. Dephosphorylation takes place after the binding is over. Another 180 degrees rotation of the carrier protein takes place, and now the potassium has come inside the cell, and in the final stage it is liberated. So here what is happening, if you notice here, 3 sodium ions are moving from inside to outside and 2 potassium ions are coming from outside to inside. Now what is the significance of this 3:2 ratio? The significance of this 3:2 ratio is that 3 positive ions move from inside to outside, while simultaneously 2 positive ions are coming from outside to inside. So there is a net loss of positive ions from inside to the outside. When there is a loss of positive ions from inside to the outside, the electronegativity is maintained. Remember, in the earlier classes during the cell membrane, it would have been made clear to you that every cell has got negativity inside the cell. The negativity is maintained is because of the proteins, high amount of proteins and negatively charged particles remaining inside the cell. And why they are remaining inside the cell is because they cannot be transported out of the cell because of the big size. So always there will be a negativity inside the cell. Once this negativity gets disturbed, it becomes important that it has to be restored. A cell inside losing its negativity means there are positive charges coming in, and this positive charges has to be moved out back into the outside so that electronegativity inside is maintained. Excuse me. So now one of the function of this we can understand from the whole of this concept is it is an active transport. It is restoring the concentration of sodium inside the cell and potassium outside the cell. Thirdly, it is also restoring the negative charges which are present inside the cell. So the electrogenicity of the cell is maintained because of the sodium potassium pump. Next, the same thing is explained here in another diagram, where if you see, this is exactly the channel, this is a cell membrane, this is the inside here, this is the outside here, this is the cell membrane. In between the cell membrane there is a channel protein, and this protein is nothing but a sodium potassium channel protein. Here it has got active sites for the sodium ions and the active sites of potassium ions. The potassium concentration is outside there, in here it is the sodium ions. You know that the sodium ions now here are less, outside more, potassium inside is more, outside is less. So whenever these three sodium ions comes and bind on these three active sites, the Atpase enzyme will get activated. Activated of this ATP enzyme will cause phosphorylation. So this binding will cause a conformational change. Because of the conformational change, these three sodium ions will go outside and potassium will come inside. As a result of which two things will happen. The concentration of sodium ions and potassium ions in the ECF and the ICF will be maintained in equilibrium, or a homeostasis will be maintained. Secondly, the electronegativity which was disturbed, restoration of the ionic potentials will be brought back to normal. So here the same diagram again for you to understand. This is again the ECF, inside you have the ICF, this is the channel protein, the gates are closed on the outside, here you have got the binding site, three sodium ions are coming and they are binding on three active sites. When these three sodium ions bind on the binding site of this sodium potassium pump, this ATP will get activated. Activation of this ATP will cause phosphorylation of ATP into a phosphate bond, and ADP and energy will be released. Once this phosphorylation will take place, immediately the active site which is closed here will get open. Once it gets open, you will find that sodium will move from inside to outside against the concentration gradient. Simultaneously, the potassium active sites will have high affinity now. So potassium will come from outside and bind onto this binding protein or the sodium potassium pump. Once the sodium are liberated, their dephosphorylation will take place. You will see here there was a phosphate bond. This phosphate bond is released. Now the phosphate bond is not there. So dephosphorylation will take place. Once the dephosphorylation will take place, a conformational change will take place. The active sites of the gates will close and the sodium ions will be released inside. So restoration of sodium potassium ions inside will happen and the sodium on the outside takes place. So this is a live example. Here you can see a small demo I have tried to put it here to explain easily, where this is the sodium potassium pump in the cell membrane. Sodium are in yellow, the potassium are in the orange color. So these three sodium ions binding on to the binding side on the inside of the cell. Once that binding takes place, energy is released. Energy released of the energy causes the bond to cause a structural change in the channel, and this structural changes in the channel causes the potassium to go outside. Once the potassium goes outside, dephosphorylation takes place. Potassium ions are binding simultaneously. Once this dephosphorylation takes place, the potassium ion now is released from the binding site on the inside of the cell membrane. Thus, the electrochemical gradient is restored back to normal. So you will find here simultaneously what is more important also is three sodiums are going out, two potassiums are coming in. Thus, there is a loss of net one positive charge. So when there is a loss of one positive charge on the inside of the cell membrane, that is one positive charge is lost from inside to outside, the electronegativity is maintained, or in other words we can say the electropositive ions or the positivity comes down and electronegativity is restored back to normal. Now, brief description of the sodium potassium pump. Here you have got two units. One is an alpha subunit and the other is a beta subunit. The molecular weights of the alpha subunit is lower one, 10,000. The beta subunit is around 55,000 daltons. The alpha subunit has got a catalytic activity. It has also the ability to bind the sodium and the potassium and a cubane. Cubane is nothing but a drug. I will come on to that in applied physiology. Whereas the beta subunit does not have any catalytic activity. It just acts as an anchor to the directly cell membrane and to the alpha subunit. So basically remember that there are two subunits, alpha subunit and a beta subunit. The alpha subunit possesses the catalytic activity, that is the breakdown of the ATP enzyme is present there, and also the alpha subunit has got the ability to bind to the sodium and potassium ions, whereas the beta subunit only acts as an anchor. To where does it anchor? It helps in binding the alpha subunit to itself and then to the cell membrane. Now, what are the functions of the sodium potassium pump? Now, this sodium potassium pump is basically found in almost all cells of the body. Literally 100 trillion cells, each of them will contain the sodium potassium pump. Now, the coupling ratio here of the sodium potassium pump is that it is 3:2. What do you mean by coupling ratio? Coupling ratio means 3 positive ions, that is sodium ions, binding on one side, 2 positive ions, that is potassium ions, binding onto the inside. So the 3:2 it is the ratio, and because of this coupling ratio, antipode direction, sodium moving from inside to outside, potassium moving from outside to inside, it alters the electrical nature of the cell activity. Hence, it is also known as an electrogenic pump. What are the uses of this pump? The major, it is the process or it is the pump which helps or which consumes most of the energy in the body. Once any activity takes place, let us take your neuronal activity. You are thinking, sitting and thinking in your brain. You are using your neurons. You may be writing down some points right now. So there is activity of neuronal activity plus muscular activity. I sitting on this side, I am speaking and talking. So there is some energy being used in the muscles of the oral cavity. Plus, any activity when you are playing games or anything requires certain energy and all the cells are being functional. So, when these cells are being functional, always there is a movement of sodium ions and potassium ions across the cell membrane. This equilibrium is disturbed because of the activity, and this disturbance has to be restored, and that restoration of this disturbance, that is the restoration of the sodium concentration outside and the potassium concentration inside the cell, is being done by the sodium potassium pump. 70% of the neurons, 70% of the energy used by the neurons is by the sodium potassium active pump. 33% is the other energies which are being used. So what is the other function? First, restoration of the electrolytes, then restoration of the electrogenicity of the cell. Third one, it also helps to regulate the cell volume. Now, to give you the example of how it helps in regulating the cell volume, let us discuss it like this. Say for example, whenever there is an entry of sodium ions inside the cell, there is water coming inside the cell from ECF. Whenever glucose, you are taking your breakfast, lunch, it is broken down into simple carbohydrates, fats. Carbohydrates are glucose. The glucose is absorbed from the stomach, enters into the circulation, and with the help of insulin, the glucose enters into the 75 trillion cells or all the cells of the body. Now, when this glucose is entering into the cell, it is entering along with sodium ions also at some times in some parts of the body, and it is also carrying along with it water. Now, if this water which has entered into the cell is causing an increase in the size of the cells, swelling up of the size cells to occur, that means the ICF volume is increasing. Now, once this ICF volume increases, the cells become swollen, and it becomes important that this water has to be pushed out from the ECF into the outside, from the ICF to the ECF. This is being done by the sodium potassium pump. So whenever there is a pump getting active, some amount of water is also restored, is taken along out from inside the cell to outside, thus the cell volume is restored. Then it also helps in maintaining the resting membrane potential and action potential. Now at this point of time, we just remember that there is a electronegativity occurring in almost all the excitable tissues of the body. The excitable tissues of the body are basically nerves and the muscles. So all this will have electronegativity inside, and this electronegativity is maintained because of the influx of sodium ions. And whenever there is an action potential, or there is some stimulus given, or an action potential is generated, this electronegativity is disturbed. To bring that electronegativity back to normal, the sodium potassium pump helps. It helps to restore the sodium and the potassium ions back to normal. In addition to this, you will also find that the sodium potassium pump is also helpful in functioning of the normal endocrine system in the body, especially the sodium ions and the sodium in the potassium pump functions well, helps the functioning of the thyroid hormones. So for a normal thyroid action to take place, we require that a sodium potassium pump has to be active. Then even for the functioning of certain insulin, for the normal functioning of insulin, a sodium potassium pump is essential. So not only it regulates the cell volume, it maintains the electrolyte concentration normal within the cell. It helps in maintaining the electrogenicity normal, that is the ionic charges are maintained normal. It also helps in the normal functioning of the endocrine system of the body. In addition to this, whenever there is a whenever the electronegativity inside the cell is normal, the protein synthesis will also take place at an optimum level. So maintaining a normal electronegativity is one of the essential factors for a normal protein synthesis to take place inside the cell. Hence, the sodium potassium pump sees to it that whenever the electronegativity is disturbed, it brings it back to normal. So indirectly, protein synthesis also is helped in the sodium potassium pump helps in the normal protein synthesis. Now, what are the factors which are regulating the sodium potassium pump? The factors which maintains the sodium potassium pump or regulates are some of the hormones. For example, the thyroid hormone will cause an increased activity of the sodium potassium pump. So it increases the pumping activity. That means if the normal sodium potassium pump works around one rotation per second or 60 times in a minute, thyroid activity can increase it to 100 times per minute. Similarly, aldosterone also increases the pumping or the activity of the sodium potassium pump. Insulin also increases the sodium potassium pump activity. So therefore, the three hormones which regulate the activity of the sodium potassium pump is thyroid, aldosterone, insulin. The other activity is also an indirect activity where the second messenger which is present binds on to the cell membrane and regulates the sodium potassium pump. Now, for example, if a hormone binds onto the cell membrane, binding up of this hormone to the cell membrane will stimulate the second messenger. The second messenger is nothing but cyclic AMP. This cyclic AMP will regulate the sodium potassium pump. Factors which will decrease the activity of the sodium potassium pump is, some of the factors which regulate or decrease the activity is hypothermia. Whenever the body temperature falls below normal, the activity of the sodium potassium pump is decreased. That is hypothermia. Then hypoxia. whenever there is a decrease in the oxygen concentration of the atmosphere, or when you are not able to breathe properly, resulting in less oxygen entering into the body, that condition is called as hypoxia. So even in hypoxia, you will find that the sodium potassium activity is decreased. It does not function normally. Third, there are certain drugs which are useful which are used to reduce the activity of the sodium potassium pump. For example, digitalis. Digitalis is one drug which directly inhibits the sodium potassium pump. Cubane is another drug which blocks the sodium potassium pump. Now, what is this sodium potassium pump? It in the heart, it reduces the force of contraction of the heart. So, sodium potassium pump is a pump where when it is present in the cardiac muscle, it helps in increasing the force of contraction. So, digitalis and other drugs will block this pump and reduce the activity of the sodium potassium pump. So always remember, certain drugs help in reduction of the activity of the sodium potassium pump. One of them is digitalis and the other is the dopamine. Now, apart from this, there are certain other transporters where you will find that the active transport takes place. One, as I told you, the sodium potassium pump is there. Then you have got the calcium ATPase pump. You have got the hydrogen potassium pump where it is in the stomach and in the kidneys. Then you have got the proton pump or the hydrogen ion pump, which is being present within the cell organelles, and in the cell organelles it helps in supplying the hydrogen ions to the mitochondria, to the lysosomes and to the Golgi apparatus. So basically today these are some of the examples of the primary active transport. In the next class we shall discuss, or we shall in brief discuss about the secondary active transport and other methods of transport. Thank you one and all. Have a good day. Another announcement which I wanted to make you is that the class, that is the anatomy class, subsequent anatomy class, for which is supposed to happen at 11 to 12 today, is cancelled because of some prior commitments. Thank you. Have a nice day.